Food resources are limited on international space stations. Thus, having a stable nutrient intake for astronauts is crucial in long-term space exploration. This is why we came out with the Astroyeast project.
This year, our team is working on space-compatible yeast for nutrient biomanufacturing. We're also building a bioreactor to help us create the ideal environment to culture the yeast in space while minimizing stress conditions. The bioreactor controls nutrients, oxygen supply, cellular waste, and temperature.
First, we plan to construct a stress reporter system with gene promoters selected last year using GFP. The system will be made within the yeast by CRISPR-Cas9 mediated homologous recombination. From this reporter system, we will get a quantifiable signal that will be used to track promoter expression changes in an induced microgravity environment. The signal will help us identify the regions that need to be modified to create our microgravity-tolerant yeast strain. In adaptive evolution experiments, cells are subjected to prolonged periods of exposure to new environments, letting them adapt due to selective pressure. For our project, we will use our reporter strain to undergo genome mutagenesis by UV radiation, which will cause random mutations creating a genetic diversity. It is at this point that we will be using our 3D clinostat to mimic a microgravity environment.
Our space-compatible yeast opens the door to many biomanufacturing applications for long space journeys. At the moment, we're also looking at the possibility of integrating genes for vitamin and flavor production.